Welcome everyone to our comprehensive guide to understanding the toxicokinetics. In our 15 minutes video, which is divided into 3 parts, we will delve into Introduction to Toxicokinetics, the fundamental processes that govern how toxic substances enter, move through, metabolized, including exit from the body, and lastly, conclusion. So, let's begin. What is toxicokinetics? Well, firstly, toxicokinetics plays a fundamental role in toxicology, as it forms the cornerstone of understanding the behavior of toxic substances within biological, human, animal, plant systems. The term toxicokinetics is derived from the combination of toxic, referring to poisons or harmful substances, and kinetics, which deals with the rates of chemical processes and transformations. Apparently, toxicokinetics originated in 1976 till 1977, when P.J. Gehring and co-workers reported the essential relevance of pharmacokinetics in evaluating the results of chronic toxicity, carcinogenicity studies in laboratory animals. Now we understand what is toxicokinetics. What about xenobiotics? The term xenobiotics comes from the Greek xenos, foreign, and bios, life, meaning foreign things in living form. Xenobiotics are defined as chemicals found but not produced in organisms or the environment. Xenobiotics are compounds of synthetic origin found in soil, water, plants, animals, and humans. They are categorized into many groups as above. Now, you might also heard of the term pharmacokinetic. Of course, you might be confused too. Well, what is the difference? Pharmacokinetics and toxicokinetics differ in terms of goals and technology. By definition, pharmacokinetic studies are conducted at pharmacologic or therapeutic doses in order to characterize xenobiotic disposition in experimental animals and humans at therapeutically relevant doses. Toxicokinetic studies are generally carried out exclusively in animals at much higher doses than those used in pharmacokinetic studies. Pharmacokinetic studies are a collaboration with pharmacology, clinical pharmacology, and clinical development. Obtain pharmacokinetic and pharmacodynamic data to provide a link between preclinical studies and humans. Toxicokinetics, on the other hand, is to quantify the pharmacokinetics of the compound in toxicology species at toxicological doses, to establish appropriate xenobiotic exposure in these species, establish metabolic profiles of new chemical entities in the toxicity species relative to humans. Basically, in simpler terms, research data from pharmacokinetics is further studied in toxicokinetic studies. In a nutshell, toxicokinetic study actually provides the basis to understand the potential health effects and risks associated with exposure to xenobiotics, including supporting the development of effective risk assessment and management strategies in areas such as pharmacology, environmental health, and toxicology. We are now moving to the principles of toxicokinetics. It is divided into 4 stages, which is absorption, distribution or translocation, metabolism or biotransformation, and lastly, excretion. The principles involves the absorption, which is how the body absorbs the toxins, distribution or translocation, metabolization, and excretion of the toxic substances, which relates to the concentration of the xenobiotic at the action site that determines the toxicity. Why, when, and how absorption happens. A known fact: absorption happens when someone inhaled poisonous gas, ingested a poisoned food or topical application, an ointment or a cream on their skin. This determines the initial concentration of the toxin in the blood, setting the stage for the next phases of its journey. Absorption is the gateway through which toxins access the internal environment of the body. The percentage of a toxin reaching systemic circulation is referred to as bioavailability. Factors determining the absorption of the toxin determines the bioavailability. For this video purposes, we focus on ingested xenobiotics. Various factors determine the rate and extent of absorption is the type of exposure route, type of membrane transport, host alimentary tract, gastrointestinal pH, motility, mucosal epithelium, intestinal microflora, lymph, and blood-brain barrier. Firstly, type of exposure. The route of exposure significantly influences the absorption mechanism. The most common pathways include oral, gastrointestinal tract, inhalation, respiratory tract, dermal, skin, and specialized administrative routes such as intravenous IV, intramuscular IM, and subcutaneous SQ. Each route has distinct barriers and surface areas affecting absorption rates. First exposure route is the oral gastrointestinal tract, as these, the most common route for many toxins, enter the body through ingestion. From the mouth, they travel to the stomach and intestines, where they are absorbed into the bloodstream. The surface area of the intestines and the presence of transporters facilitate or hinder their absorption. Second most common exposure route is inhalation, respiratory tract, for gases, vapors, and particulate matter can be inhaled into the lungs, where they diffuse across the alveolar membrane into the blood based on their concentration gradient. The large surface area of the lungs facilitates rapid absorption of inhaled substances. Thirdly is dermal, skin, whereby some substances can penetrate the skin and enter the bloodstream. The rate of dermal absorption depends on the skin's integrity, the substance's solubility, and its ability to overcome the skin's barrier function. And lastly is intravenous, intramuscular, subcutaneous, which involves the direct injection into veins, muscles, or under the skin, offers almost immediate absorption, bypassing most natural barriers. Secondly, type of membrane transport. The physicochemical properties of the toxin, such as molecular size, solubility, water versus lipid, and ionization state, affect how easily a substance crosses cell membranes. Nonpolar or lipid-soluble substances tend to be more readily absorbed than polar or ionized substances. An increase in droplet size increases dose too. As for xenobiotic concentration and dose, the higher the concentration and dose, it increases the absorption rate. However, the presence of food or other substances can interact with the xenobiotic in the gastrointestinal tract, either enhancing or inhibiting absorption. The movement of the chemical across the cell membrane also influences the absorption, using passive transport or active transport. Passive transport or passive diffusion includes simple diffusion and facilitated diffusion. They do not require ATP energy and are driven by concentration gradients such as ethanol. Facilitated diffusion is a carrier-mediated transport of water-soluble substances that mimic the structures of endogenous substances that normally exist in the body. For example, glucose normally enters cells via special glucose transporters. Active transport is a carrier transport through membrane against a concentration gradient which requires metabolic energy. As with the glucose-facilitated diffusion transporter, toxic agents with structures similar to a nutrient can compete with the nutrient for active transport into the cell. An example is heavy metals such as lead may enter through the transporter that normally carries calcium. Third, host alimentary tract. The xenobiotic's molecular size and lipophilic characteristics absorption rate depends to the physiological, anatomical features of each section, residence time within each section, and presence of food. The highly acidic stomach pH favors the absorption of weak acids because it is more lipid-soluble in their non-ionized form compared to weak bases or ionized forms. Small intestine is the principal site of absorption of dietary xenobiotics, with pH ranking from 6 to 8, at the proximal small intestina and distal ileum. With the long length and highly absorptive tall columnar cell enterocytes, absorption of weak acids and bases is nearly complete. In addition to this, the presence of food also reduces the bioavailability of orally consumed xenobiotics in food. Since intestinal have larger surface area, due to the presence of villi and microvilli, xenobiotics are absorbed more there compared to the stomach. Presence of gastrointestinal motility from the action of smooth muscle in the intestinal walls produces tonic contractions. This increases the concentration of diffusible xenobiotic at the site of movement. The permeability and thickness of the mucosal epithelium with the intestinal blood flow helps to maintain the concentration gradient across the mucosal epithelium, which is capable of rapid transfer of the xenobiotics across the epithelium. The unique feature of the intestinal tract is that it has diverse microbial community, mostly anaerobic. Intestinal bacteria mediate the metabolism of these xenobiotics that may increase or decrease their bioavailability. This bacterial metabolism activities such as reductions, hydrolyses, and degradation results in individual variations in sensitivity to many orally ingested xenobiotics. Understanding the absorption process is crucial because it determines toxic effects, Analyses the risk assessment and management, including treatment strategies. The degree and rate of absorption influences the onset, intensity, and duration of a toxin's clinical sign. There are also other factors affecting absorption of the xenobiotic, which affects the efficiency and speed of absorption. Besides the route of exposure, which physicochemical properties of the toxin, type of transport, and xenobiotic dose, as well concentration. Next, lymph. There is a great tendency for lipophilic xenobiotics to be associated with dietary lipids and intestinal lipoproteins, chylomicrons, that results in their selective absorption in the lymph system. Non-lipophilic xenobiotics tend to be diverted toward the blood system, which has a higher blood flow versus the slow lymph flow. So, after all the determining factors affecting the absorption of the xenobiotic in the gastrointestinal tract, the xenobiotic enters the lymph that drains into the thoracic duct and the portal vein draining into the liver. At the liver, the xenobiotic are mostly completely removed from the blood by hepatocytes during the first passage of blood through the liver known as first-pass metabolism. As a result of this, the xenobiotic might not reach systemic circulation in high concentration. Thereafter, the xenobiotic enters the central vein along the liver sinusoids to reach the systemic circulation for distribution from the central, blood, to peripheral tissues, including the site of toxicity action. Once absorbed, toxins distribute throughout the body, transported by the bloodstream. Since not all tissues are created equal, the distribution is influenced by blood flow, tissue permeability, and the toxin's affinity for fat versus water. Highly vascular with higher blood flow in organs like the liver, kidneys, muscle, brain, and skin often receive more of the xenobiotic substance compared to bone and fat. The venous blood flow from the liver is pumped by the heart to the lung for oxygenation. Then arterial blood is pumped directly to all organs, where blood is redirected again to the heart through the venous blood. Blood returning from the gut enters the hepatic portal veins and flows to the liver. So, the liver metabolizes any xenobiotic before it enters into the general circulation. The abdominal lymph flows into the thoracic ducts, which then is united with the blood at the left jugular and left subclavian vein immediately before it enters the heart. Xenobiotics absorbed from the small intestines will avoid first-pass metabolism in the liver and encounters the less efficient xenobiotic metabolic apparatus of the lung. The bile produced in the liver contains xenobiotic metabolites, is transported to the upper small intestine, duodenum. These metabolites are exposed to the enterocytes enzymes and gut microbes, which is then reabsorbed again into the liver. This recycling process is called the enterohepatic circulation, which increases the residence time of the xenobiotic in the body. The distribution or translocation is the interorgan movement of xenobiotics depends to the distribution volume and plasma protein binding. This both affects the xenobiotic agent biological effects. Xenobiotic bound to plasma proteins cannot cross capillary walls due to high molecular mass, thus decrease the toxic effects of the xenobiotic, but it is an efficient means for transport for xenobiotics to various tissues since this association is reversible. Some xenobiotics tend to accumulate in tissues at higher concentration due to active transport or nonspecific binding. The tendency for xenobiotics to be stored in the body is because of their polarity. For example, the adipose tissue can store fat-soluble substances for longer periods. Inorganic substances tend to bind to non-selective sites in bone and selective sites such as kidney and liver. Understanding distribution is crucial for pinpointing where a toxin might exert its effects and cause damage. Storage sites that are not target site will not reduce toxicity potency, but if the storage at the target site, it will increase toxic potency. The slow release from storage site result in chronic toxicity. Factors affecting the xenobiotic distribution are divided to physiochemical properties, blood flow to tissue, binding to macromolecules, and anatomic barriers. In physiochemical properties, non-ionized xenobiotic is more lipid-soluble, thus penetrate intracellularly. While highly polar, lipid-soluble, xenobiotics confined to interstitial space and xenobiotics with low molecular weight move freely through the capillary endothelium. Tissues or organs that has high blood flow achieve greater xenobiotic concentration compared to poorly vascularized organs and vice versa. Likewise, unbounded and free can perfuse the endothelial cells and enter to the target site. This also allows tissue binding at the site, and if it is the target site, effects of toxicity can be seen. However, some organs or tissue have barriers such as tighter and small junctions in blood-brain barrier, eyes and testicles. This poorly vascularized organ and their barriers limits the perfusion of xenobiotics unless it's lipid-soluble. Metabolism or biotransformation of toxic agents by the body is an attempt to detoxify. In some instances, metabolized xenobiotic agents are more toxic than the original compound. This is referred to as lethal synthesis. The concept of metabolism is to increase the water solubility of the xenobiotic, so it can be excreted via kidneys. Most xenobiotics are eliminated by hepatic metabolism, renal excretion, or both. Lipid-soluble xenobiotics are converted to water-soluble form before they can be eliminated by the kidney. There are two phases of metabolism. Phase 1, metabolism chemically changes the xenobiotic so that it is more water-soluble and more susceptible to Phase 2 metabolism. Phase 1 reaction includes oxidation, reduction, and hydrolysis mechanisms. These reactions are catalyzed by hepatic enzymes known as cytochrome P450 enzyme. Other organs with this enzyme are lungs, skin, and kidney. Phase 1, metabolites can be inactive, less active, or more toxic than the parent compound. Products of Phase 1 may be excreted if polar solubility permits translocation. Phase 2 principally involves conjugation, when a large water-soluble molecule to either parent xenobiotic or a Phase 1 metabolite. Common conjugates include glucuronides, glutathione, and sulfation. Metabolism of xenobiotic agents seldom follows a single pathway. Usually, a fraction is excreted unchanged, and the rest is excreted or stored as metabolites. Every individual has their own genetic diversity. There is genetic polymorphism exists for the CP450 superfamily enzymes. Some xenobiotic agents may inactive or activate the CP450, such as organophosphate. In general, older animals and neonates have lower oscillativity of all hepatics biotransforming enzymes. Additionally, severe liver disease or chronic liver failure also compromise the liver activity. If the xenobiotic is characterized by a high extraction ratio, it will limit the update for other metabolites, since hepatic biotransformation are saturable with limited capacity, especially when the amount of xenobiotic agent presented to the liver exceeds its capacity. Also, the greater the protein binding, the slower the rate of metabolism. Excretion, which is the body's way of eliminating toxins. Xenobiotics, or toxicants metabolites, are excreted via urine, feces, exhaled air, saliva, perspiration, and milk. Kidneys play a starring role here since it is a primary route of toxicant excretion. It filters blood and removes waste through urine. Nephron, the kidney functional unit, is divided into glomerulus, proximal convoluted tubule, and the distal convoluted tubule. The excretion of urine involves three processes, namely filtration, secretion, and reabsorption. In fecal elimination occurs in two processes, which is the excretion in bile and direct excretion into the lumen of the gastrointestinal tract. The biliary route, also an important mechanism for fecal excretion of xenobiotics and their metabolites. The biliary route is an effect from the enterohepatic cycling. However, that being said, intestinal excretion is a slow process, and therefore for xenobiotics that have slow rates of metabolism or slow rates of excretion by other means. In conclusion, the journey of toxins through the body is a complex dance of absorption, distribution, metabolism, and excretion. Each step interlocks with the next, illustrating the body's intricate mechanisms to protect itself from harm. We hope this video has provided you with valuable insights into the intricate mechanisms of toxicokinetics. Thank you for watching, and we encourage you to continue learning about this critical aspect of toxicology.